Today we're going to begin a discussion of cell division and reproduction. Before we actually, and today we're actually working on a type of division called mitosis. But before we get into the details of mitosis, I first want to talk about something called ploidy, and it's a term I'll probably be using quite a bit. So ploidy is a reference to the number of sets of chromosomes that a particular cell has. And so some of the terms that we use here, one of these is haploid. So the term haploid means that a cell has one complete set of chromosomes. And so if we use the example of humans, we have 23 distinct and different types of chromosomes. And by that I mean chromosome number 1 would be coding for different information than chromosome number 2, and that would be coding for different information than number 3, from number 4, all the way through 23 chromosomes. So if a cell had one set of 23 chromosomes, we would call that cell a haploid cell. And in our bodies, and this is typical of animals, really the only cells that are going to be haploid will be our sex cells. So sperm and eggs would carry one set of chromosomes. And the rest of our cells, as it turns out, is going to have two sets of chromosomes. So another example of ploidy then would be cells that do have two sets of chromosomes, and we call those cells diploid cells, and that's represented by 2N. So again, in our cells, in our body cells, everything but our sperm and eggs, we have two sets of chromosomes. So chromosome number 1, we would have 2 of chromosome number 1, 2 of number 2, 2 of number 3, number 4, all the way through 23. So where we get these sets of 23, we get one set of 23 from mom, one set of 23 from dad. So one set of 23 in sperm, one set of 23 in the egg. And when they fuse together, that establishes a cell that's diploid. And they will have different characteristics, potentially. So we refer to these sets of chromosomes or these pairs of chromosomes as homologous pairs. So again, we would have 2 of chromosome number 1, meaning that we get one chromosome number 1 from dad, one from mom. Now, they will have the same general traits. So perhaps this codes for eye color and hair color. So maybe dad has blue hair and blue eyes, and mom has red hair and red eyes. The point is that the chromosomes code for the same general traits, but they may not be exactly the same because mom and dad may have different genes for those particular traits. So again, in a human cell then, a typical body cell then, we have 2 sets of 23, so most of our cells are diploid. Now, some organisms can have more than 2 sets of chromosomes, and we refer to those kinds of organisms as being polyploid. So the prefix poly means many, so many sets of chromosomes. So this is unusual in animals. Normally when we talk animals, we're talking diploid and haploid, and certainly as we talk humans, we'll be talking diploid and haploid. But plants, for example, can tolerate being polyploid pretty easily. So in this example, you're seeing that there are 4 number 1s, 4 number 2s, 4 number 3s. So that would be a 4N cell or tetraploid cell. And again, it can be highly variable depending on what type of organism we're talking about. But polyploidy is fairly rare in nature. Most typically, we're looking at haploid, diploid cells. Okay, now we're going to be talking about a type of cell division today called mitosis. And in mitosis, what we're going to be talking about primarily are diploid cells. So we're going to start with a diploid cell, most typically. And at the end of the process, we'll end up with 2 diploid cells. And these cells are known as daughter cells. The original cell is called the parent cell. Now, one of the things to understand about mitosis is that we start with one cell. We're going to end up with 2 cells of the same ploidy. And the 2 cells will be genetically identical to one another and genetically identical to the parent cell that gave rise to them. Now, to do that, what has to happen is that first the chromosomes have to be replicated, and I'll get back to that in a second, and we've talked about chromosomal replication before. So we're going to have to take our 46 chromosomes, copy them so that we have double that, and then they'll be separated into 2 identical sets of 46 chromosomes, and then the cell will be divided in 2. So it involves 1 round of chromosomal replication, separation of those, and separation of the cell into 2 cells. So again, start with 1 cell, end up with 2. The ploidy will stay the same. So if we start with 2N cell, we'll end up with 2N cells. Along the way, we double the number of chromosomes, separate them, and then divide the cell in half. So again, those are the key components of mitosis, and we'll talk about the details here in just a couple of minutes. All right, so when we look at mitosis, then mitosis has certain functions in nature. And so one of the functions is reproduction. So if you're a one-celled organism and you go from one cell to two cells, you've just doubled the number of organisms. And so that would be an example of a type of reproduction. And that's known as an asexual reproduction, Now, other organisms can also asexually reproduce, even multicellular organisms can. So some simple animals can do this as well. For example, this is something called a hydra. Anyway, the hydra itself can actually grow a new little hydra off the side that will eventually pinch off at the base and float away. And that's a type of reproduction, but it's considered asexual because you don't need a mate. So it's without sex in effect. And plants are also very capable of asexually reproducing. For example, aspens. We used to look at aspens and we would have called this group of aspens a stand of aspens, assuming that they all developed from different seeds. But as it turns out, we would now call this a clone of aspens because they would all actually share the same root system and they generate new trunks out of the same root system through mitotic division. So in the hydra I showed previously and also with this aspen then, they are reproducing without a mate and they are reproducing asexually. Now, the good aspect or the positive aspect of asexual reproduction is that you don't need a mate, so it's easy to do. And the downside is, though, that the new organism, whether we're talking about the hydra a second ago or the new aspen tree here, would be genetically identical to the original tree or to the original organism. So asexual reproduction is good in some ways because, again, you don't need a partner. But the downside is that all of the progeny are genetically identical. And so you have very little genetic variation or diversity in the population. All right, now other functions of mitosis is certainly another one would be for growth. And we could even consider the trees for that as well in ourselves. So in other words, one time we were a single cell. We were a fertilized egg called a zygote. And that zygote began to divide mitotically and eventually produced the trillions of cells of which we are composed. And they've organized themselves and differentiated and so on. But the point is we got from that single cell, that zygote, to the many cells through mitotic division. So what that means then is that all the cells of our body are genetically identical, save for mutation. And that is true. And we've talked a little bit about that before as well. Then a third function of mitosis is tissue regeneration. So if tissues are damaged or if an organism is damaged, mitosis will be used to produce new cells to replace the damaged tissue. So we certainly have, as humans, have some capacity for regeneration. If we damage our skin, we scrape our skin, we can grow new skin. If we break a bone, we can generate new bone to replace that. And again, that would all be done through mitotic division. But we also have limitations. It's not as if we can lose a limb, lose a hand, and regrow a hand. So we have some capacity for regeneration, but not complete capacity for regeneration. But again, that would all those cells would be replaced through mitotic division. Okay, now, when we look at mitosis, then mitosis is part of a larger process, which we call the cell cycle. And so in the cell cycle, the actual division occurs in what we call mitosis. And then there's another phase that we refer to as interphase. And an interphase cell is a normal-looking cell through the light microscope. And mitosis, we actually see physical changes in the cell that eventually result in two cells being produced. Now, within interphase, there are certain subphases. So immediately following a division, a cell will go into what's known as gap phase 1, G1. And in gap phase 1, what happens is that the cells enlarge to their normal size. Again, following the previous division, the cells may have pinched off, and so it's only half the appropriate size, so it'll enlarge to the appropriate size. Organelles may need to be replaced, new ribosomes generated, things like that. So this is a process of getting back to normal, the G1 or gap phase 1. Then depending on the nature of the cell, a couple of different things can happen. Some cells will go into what we call G0 or gap phase 0. And this is where the cell will just go about the business of being a cell. So for example, if a liver cell just divides to produce new daughter cells, they may go into the business of being liver cells. Now, embryonic cells, basically their job is to just divide. But the really key phase of interphase is this next phase, which is the S subphase, which stands for the synthesis phase. And this is where the chromosomes are replicated. And what you should know about the S subphase is that once a cell enters the synthesis phase, division will follow. Mitotic division will follow. So that's the key phase. Once you initiate replication of chromosomes, you're going to end up with two cells at the end of this. So again, we're looking at a number of different cells here. These are actually onion cells. And if we look at these two cells right here, this just follows a previous division. And so these cells would be in gap phase 1. So this just follows the previous division. They're in gap phase 1. And then, again, they would either go into the business of just being onion root cells, or they may actually enter the synthesis phase, which is where replication of chromosomes begins. So again, gap phase 1 follows the previous division, either into gap phase 0 or S subphase, which really is the beginning of initiating division. So looking at the synthesis phase, we've talked about replication of chromosomes before. And so again, we would start with one chromosome, 2 strands of DNA. And again, we've talked about the role of DNA polymerase and primase, how the 2 strands will be separated, complementary strands will be built in, and we discussed that back in Module 13. Now, what we didn't discuss is that at the end of that process, the replicate chromosomes will be connected by a structure known as a centromere. And so I've exaggerated that here in red. And while a chromosome is attached to its replicate, we refer to those as sister chromatids. So this is a place where I kind of diverge a little bit from the terminology and really how this is presented in most textbooks. In that what your textbook would say, or what a textbook would say, is that this is a chromosome composed of 2 sister chromatids. But really, the sister chromatids are new chromosomes themselves. So it's kind of an absurdity to really describe it that way. So when I use the term sister chromatids, what I want you to understand is that sister chromatids are chromosomes that are attached to their replicate by a centromere. So rather than have to say that whole phrase, a chromosome attached to its replicate by a centromere, I'll just be using the term sister chromatids. But the reality is that the sister chromatids are 2 chromosomes. And again, they're just attached to one another. So again, we'll be looking at some other phases later, but again, this would constitute one sister chromatid. Here's another sister chromatid, and they are attached by a centromere. So these are sister chromatids, each one really a separate chromosome. And again, same idea. We've got sister chromatids here and here attached by centromeres, here and here. And again, if we wanted to uncoil that, we'd eventually see the proteins and DNA of which they're constituted. Okay, so again, the idea here then is that diploid cell doubles the chromosome number and is eventually going to separate those chromosomes into 2 identical halves and then divide the cell in 2. And that's why we end up with 2 identical chromosomes at the end of the process. So immediately following the previous division, cells go into gap phase 1, where they basically reestablish their normal size and organelles. They may either just go into the business of being that particular type of cell, or they may decide to divide again. If they enter the S subphase, that means they are going to prepare for division. Now, the other thing I want to mention is that since we are in interphase, if you were looking at the cell with the light microscope, you would not know that the chromosome number has doubled. You can't tell by looking. You'd have to actually measure the DNA to know that that's the case. And then after the chromosomes have doubled, the cell will enter gap phase 2. And gap phase 2 is where the cell begins to accumulate the proteins it's going to need for division, and also centrosomes will replicate as part of this process as well. And so again, the centrosomes are used and help build some of the machinery that are needed for cell division. And so that would be part of interphase as well. And again, you can't really tell by looking at the cell that that's happened with the light microscope, but that is the end of interphase. So I want to use a couple of visuals then to show us what's happened at the end of interphase as the cell is ready to enter true mitosis. So I'm going to use the Elmo here, the overhead camera, for just a second. And so if we imagine a nucleus, and let's imagine this is a diploid cell. And so these clay models, and this is a really good way to learn mitosis, by the way, is just get some clay or get some Play-Doh and actually work through it. But this is going to represent a diploid cell. So the blue is representing chromosome number 1, but it's also representing 23 chromosomes inherited from dad. And this is representing 23 chromosomes inherited from mom. And so what's going to happen in the S subphase of interphase is that all of dad's chromosomes will replicate and be attached to the replicate by the centromere. And all of mom's chromosomes will replicate and be attached to one another by the centromere. Okay? So that will get us through interphase, and then in gap phase 2, centrosomes will replicate. Now I'd also like to show you an animation as well that will show us some of these events of interphase, and then we'll continue on in mitosis. So if we look at interphase, here we're looking at the cells of interphase moving into the cell and into the nucleus. And so we're seeing the chromosomes here, very thread-like, hard to see, and they now will replicate. And of course, at the end of this process, then, we would consider them sister chromatids because the chromosome and the replicate will be attached by a centromere. All right. Now, I think I'm going to go ahead and stick with the animation because that's going to take us into the next phase. But to be honest with you, actually, I do need to come back to our slides. And so what I want to do is kind of summarize where we are and where we're headed in terms of mitosis. So we've talked about interphase, gap phase 1, gap phase 0, synthesis subphase, and gap phase 2, where the centrosomes are replicated. And now where we're headed is into mitosis. So I want to quickly go through the terminology, and then we'll work through these phases. So mitosis is where the cell actually now begins to look different through the light microscope. So prophase, chromosomes begin to coil and condense, become visible. Also, the machinery for division of genetic material begins to become established. Metaphase, we'll see the sister chromatids line up at the equator of this so-called spindle. We'll see separation of the chromosomes in anaphase. And these three phases together generate karyokinesis. Karyokinesis is the division of the nuclear material or separation of the chromosomes. And then anaphase is followed by telophase, the so-called end phase. And in telophase, that's where the actual cell division takes place, and that's called cytokinesis. So that's where we've been. That's where we're headed. All right. So I'm going to work through, first I'm going to go to this animation, and then I'm going to come back to my Play-Doh models, and we'll look at these phases with some extreme differences in visuals. So let's go back to our animation, and we'll pick up where we left off. So the chromosomes have been replicated. And again, if we were looking at the cell, it would be a normal-looking cell. So next, what begins to happen then is that the chromosomes begin to coil and condense. And when we're looking at these chromosomes, or I should say sister chromatids through the light microscope, they become visible. So previously we couldn't really see them, but as we look at the nucleus now, the nucleus is becoming more granular and the chromosomes are becoming distinguished. And so we call this phase prophase. Now, as prophase continues, again, what will also have happened, remember in the G2 phases, the centrosomes will have replicated. And a structure is being produced called the mitotic spindle. And the function of the mitotic spindle is to separate the sister chromatids from one another. So again, the coiling and condensing of chromosomes will take place in prophase. And they become visible. We'll be able to actually see the chromosomes through the light microscope, and I'll show you some slides of that in a second. And in addition to that, the spindle is being produced. And the spindle have fibers, some of which will attach to the centromeres, and some of which run the entire length of the spindle. And so again, these microtubules are utilized to help move chromosomes around. And so you can see that some of them attach to the centromeres that are attaching the two sister chromatids. So all of this is occurring in prophase. So prophase is kind of an extensive phase, and your textbooks, some textbooks are also describing this as a prometaphase, but I don't really care for that term. So again, as we kind of look at the changes in prophase then, chromosomes coil, condense, become visible, the nuclear envelope disintegrates, mitotic spindle is generated. These are the events of prophase. And if we come back over to the overhead camera then, what I think I'll do is move my Play-Doh models to a slightly more advanced phase. And so again, what would happen here is that the mitotic spindle would be produced, and our chromatids would be attaching to microtubules, and will eventually be positioned at a particular place in the equator, or so-called center, of the mitotic spindle. So once the chromatids are lined up at the center of the mitotic spindle, we refer to that as metaphase. And so again, in our diagram, we can see here that this would be the metaphase cell, all the sister chromatids lined up at the equator. And if we move back over to our animation, again, looking at what the metaphase cell looks like. So we're still in prophase here because the sister chromatids are being positioned. But once they're all lined up in the central portion of the mitotic spindle, that's the end of prophase, and that is metaphase. The next phase then is anaphase. And so what happens in anaphase is that the centromere, a portion of the centromere called the kinetochore, will replicate. And that allows microtubules to pull the sister chromatids to either end. Now, once they're separated now, they are chromosomes. So chromosomes will be pulled to either end of the cell. And once they stop moving, that's the end of anaphase, and that becomes telophase. And in telophase, the cell will actually divide in two. And we refer to this as cytokinesis. So at the end of cytokinesis, then, we're going to have 46 chromosomes at either end of the cell, and they are now daughter cells at this point, and each daughter cell is back into gap phase 1 of mitosis. So I would like to take us through anaphase and telophase also with our overhead camera then. And so one of the things I do want to point out is that even though these are homologous chromosomes, they really have nothing to do with one another during mitosis. And so again, lined up at the equator, that's metaphase. The centromere kinetochore replicates. That allows the opposing forces of the microtubules to separate the sister chromatids. Once they're separated, they are considered chromosomes. And so for each of the 23 chromosomes then, or what were sister chromatids, one goes to one end, one goes to the other. And I should actually say that would be for all 46 of them. One goes to each end, so we end up with 23 pairs at one end, 23 pairs at the other, and then the cell divides, and we have two genetically identical cells. Now, looking at this again through the light microscope and looking at some of these phases, just what they look like through the light microscope. Again, here's diagrammatically prophase. But in a plant cell, again, this would be early prophase. Chromosomes are just becoming visible. Late prophase, they're quite visible. Nuclear envelopes beginning to disintegrate. In an animal cell, again, these would be interphase nucleuses, fairly normal-looking nuclei. And then in prophase, chromosomes are becoming visible. In metaphase in a plant cell, we're going to see the sister chromatids lined up at the equator of the spindle. The spindle isn't quite that distinct. In an animal cell, again, we can see the chromosomes lined up at the equator of the spindle here. And then anaphase, the sister chromatids are separated, chromosomes pulled to either end of the cell. And I should mention then that the so-called kinetochore, what it actually does is kind of crawl along the microtubule, and then the microtubule disintegrates behind it. So it's not so much that the microtubules are pulling themselves apart as it is that the centromeres are crawling along the microtubules. And again, in an animal cell, this is what early anaphase would look like. And then also telophase in plant cells begins at the center of the cell. There'll actually be what's known as a cell plate. The beginning of a cell wall will be constructed across the mitotic spindle, and that will be extended to either side, and eventually the cell wall will be completed, and then we've completed telophase, and they would be daughter cells at that point. And in telophase in an animal cell, again, once their chromosomes are pulled to either end, what happens is that the cell membrane begins to pinch in from the outside, and this is again a cytokinesis. And eventually, when cytokinesis is complete, we are now daughter cells, and each of these are back into interphase, which will be gap phase 1 of interphase. So again, there's a sequence of events that lead from prophase, early prophase, late prophase, to metaphase, anaphase, and then telophase, which is where we get cytokinesis. And once telophase is complete, once the cytokinesis is complete, these cells are back in interphase, G1 of interphase. Now, something else I want to mention real briefly is that at metaphase, this is an opportunity to get a very good look at chromosomes. And so there's something called a karyotype, which is typically done of metaphase cells, cells that are dividing. And so this is one way where you can look at, say, embryonic cells, and this is typically done through a process known as amniocentesis or chorionic villus sampling. What will happen is that physicians will obtain embryonic cells by either taking a portion of the placenta or getting cells from the amniotic sac. They'll catch these cells dividing, and then they'll take a picture of the cells dividing and separating out at metaphase. And so then what they can do is go ahead and look at all of the chromosomes, see how many chromosomes of each type there are, and make sure that there are no gross abnormalities of the chromosomes and that there are the appropriate number of chromosomes. So here, for example, there's 2 of chromosome number 1, 2 of chromosome number 2, 2 of chromosome number 3, and so on. If we look at this karyotype, looks pretty normal through most of it, but if you come down here to chromosome number 21, you'll notice that there are 3 chromosomes numbers 21, and so this is what Down syndrome is, what's so-called trisomy 21. So we'll talk more about how some of these chromosomal abnormalities can occur in our next module. So, real briefly, what I want to do then is quickly kind of summarize what we have then in terms of mitotic division. And in mitotic division then, we start with a diploid cell. So in our case, we have 46 chromosomes. Those chromosomes will replicate. They'll all line up at the equator in metaphase. And then the sister chromatids will be pulled to either end. And so each cell then has 46 chromosomes. And then following the cell division, those 2 cells are genetically identical. So it's really important to get a basic foundation of mitotic division. Make sure you review this, make sure you're comfortable with the terminology, the characteristics of each phase. Because in our next module, we're going to look at a different type of cell division called meiosis. And in meiotic division, we're going to go from that diploid number of 46 to a haploid number of 23. So we're going to be looking at sexual reproduction, where we have our 2 sets of chromosomes, and we only get to pass on 1 set to the next generation. So that's where we're headed. How do we get from 2 sets of chromosomes to 1 set of chromosomes?